Zebedee, Zobiah. Okay, good morning, class. Hope we had a great night. And, it's a sunny day, and at least the sun rays are basking. I'm basking in the sun rays, and I hope wherever you are, you're feeling good and fresh. So we'll try to see we could wrap up term 2 with this. So wherever we'll stop, we would continue with it. Okay, it's gonna be a brief class, very brief. And so we're looking at the defense mechanisms of the body. The defense mechanisms of the body, and so we would get on right away. The assignments I give, please make sure you always send them in time. Okay, if they are sent after the due date and due time, they will attract negative marking, as well as it may not even, the submission may actually even be a challenge. Okay, and then if you have anything that is disturbing in submissions, don't contact me, contact the class rep. Okay, imagine me dealing with a throng of students. So look at the defense mechanisms of the body. I'll take it slowly. And sorry, please let me discharge this. So I'll take it slowly today. And so we're looking at defense mechanisms of the body, and the body is protected against pathogenic organisms by two mechanisms. Okay, the first one is phagocytosis of invading organisms, and the second one is protected against pathogenic organisms by two mechanisms. Take note, the first one is phagocytosis of invading organisms, and the second one is what? Formation of antibodies. And as phagocytosis of invading organisms, this is carried out by the white blood cells and the reticuloendothelial system. Okay, so the phagocytosis is carried out by the white blood cells and the reticuloendothelial system. And then the second one is formation of antibodies against the invading organisms, or destroying them by lymphocytes. Okay, so we have two mechanisms, two broad mechanisms: phagocytosis of the invading organism by leukocytes, by white blood cells, and the reticuloendothelial system. And then the second mechanism is what? The formation of antibodies against the invading organisms, or destroying them by lymphocytes through what is called the immune response. Okay, through what is called the immune response. Okay, so let's look briefly and quickly on the white blood cells. Remember, we've mentioned this defense is offered by the white blood cells, okay, and the reticuloendothelial system. So let's look briefly at the white blood cells. Okay? These are large nucleated cells that constitute mobile units, okay, of the body's defense mechanism. Okay, they are large nucleated cells that constitute what? The mobile unit of the body's defense system. Okay, so they seek after the organism, they seek after the invading organism, and they destroy the foreign invader. They seek and destroy. A large nucleated, okay, they are large nucleated cells, okay, they constitute the mobile unit, okay. We'll come to that, Lucas, okay. It was just an introduction, okay. It was just an introduction. We'll come to that in detail. That there are two mechanisms: the phagocytosis and formation of antibodies. Okay. So looking at the white blood cells, so these are large nucleated cells that constitute what? The mobile units. Okay. And the total count which we gave before, we said the total count was what? It's 4,000 to 11,000 for Caucasian value, and then we also mentioned the African value, right? And we can see have reasons for what we mentioned about the African value. We said the Caucasian value is about 4,000 to what? To 11,000. Okay, so we have, and then they are classified. They can be classified as what? Granulocytes and what? Non-granulocytes. Okay, some books will say polymorphonuclear leukocytes. Okay, and non-granulocytes. Okay, so for the granulocytes, they have granules in their cytoplasm. Okay, and so we have neutrophils, we have eosinophils, and we have what? Basophils. Okay, we have neutrophils, eosinophils, and what? Basophils. Okay, if you classify them in terms of the granulation of their cytoplasm, we have neutrophils, okay, or macrophages. So neutrophils, or what? White blood cells, that they consider about 50 to 70% of the white blood cell of the total white blood cell count. Okay, so I say neutrophils, or what? Macrophages. Neutrophils, or what? Macrophages. So they constitute about 50 to 70% of the total white blood cell count. We have basophil, which is about 0.4%. Okay, then we have the non-granulocytes. These ones do not have granules. Okay, they do not have granules on their in the cytoplasm. So we have the lymphocytes and the monocytes. Okay, the lymphocytes constitute 20 to 40%, monocytes 2 to 8%. Okay, 2 to 8%. So let's look at formation of leukocytes, what we call leukopoiesis. Okay, formation of leukocytes, what we call what? Leukopoiesis. Okay, so all the granulocytes and the monocytes, as well as a few lymphocytes, are formed in the bone marrow, while most lymphocytes are formed in the lymphoid organs. Okay, the granulocytes, the granulocytes and monocytes are formed where in the bone marrow. They are formed where in the bone marrow, while the lymphocytes are formed in the lymphoid organs, especially the lymph nodes and the spleen. So the examples of lymphoid organs, especially what the lymph nodes and the spleen. So leukopoiesis, just like we had for erythropoiesis, will require dietary factors that are necessary for cell growth and division. Okay, we mentioned vitamin B12, protein of high quality, of high biological value. Okay, and then this leukopoiesis is stimulated by colony, by certain colony stimulating factors, okay, that are produced by the macrophages, the fibroblasts, the endothelial cells, and certain lymphocytes. So leukopoiesis, the formation of leukocytes, is stimulated by certain colony stimulating factors, okay, and these colony stimulating factors are produced by macrophages, by endothelial cells, by fibroblasts, and even by products of inflammation. Okay, they are, and these colony stimulating factors act locally in the bone marrow, and each of the colony stimulating factors now the proliferation of a particular committed stem cells. Okay, okay. So let's quickly look at the lifespan of and fate of the leukocyte. What is the lifespan and fate of the leukocyte? What is the lifespan and the fate of the leukocyte? So the lifespan of granulocytes in the circulation is about 4. The tissues will stay for about 4 to 5 days, then they are destroyed. Okay, and some of them could be lost from the body via the gastrointestinal tract. Okay, so the lifespan in circulation is about 4 to 8 hours, after which they enter the tissues and delay there or stay there for like 4 to 5 days, and then they are destroyed. Okay, infection because what they are attacked and destroyed by the invading organisms. Okay, then the lifespan of monocytes is about 20 to 72 hours. Okay, the lifespan of monocytes is about what, 20 to 72 hours, after which they enter the tissues and enlarge to become what we call tissue macrophages. So the lifespan of monocytes, what, 20 to 72 hours in blood, please, and after which they enter into the tissue, stay for longer hour, for longer time, okay, for a longer duration or a longer period, for like 2 months or more. Okay, 2 months or more. Okay, then what about large lymphocytes? Okay, these large lymphocytes mature into smaller lymphocytes, which now reach the bloodstream via the lymphatic vessels. Remember, we mentioned that the lymphocytes are mostly produced from what? From the lymphoid organs. Okay, the lymphocytes are produced most from where the lymphoid organs. Okay, and they reach the general circulation via the lymphatic vessels. Okay, then after a few hours, they cross the capillary walls back into the tissues. Okay, from which they now re-enter the lymphatic vessels and circulate again. Okay, so they live relatively long periods. Okay, lymphocytes, they live relatively long periods. Okay, then let's look at the functions of leukocytes. Okay, let's look at the functions of leukocytes. The first one is the what, the neutrophils. Okay, we said another word for the neutrophils is what, microphages. Okay, I think I spelled that before, microphages. Okay, this, okay, the neutrophils, we said they contribute about 50 to 70%. Okay, they contribute about what, 50 to 70% of the total white blood cell count. Okay, so let's look at the functions of the neutrophils. Please pay keen attention, please. And they exert powerful defense action against bacterial infection. So each time you hear of neutrophils, what first of all comes to your mind is bacterial infection. Okay, so their main function is phagocytosis and destruction of invading bacteria. So they phagocytose, and then what, they destroy the invading bacteria. Okay. So what happens when an infection occurs, when a bacterial infection occurs? The macrophages that are present in the tissues. Remember, we have tissues, macrophages that are present in the tissues. We have fixed macrophages and we have wandering macrophages in the tissues. So, and when there's an infection, okay, the macrophages that are present in the tissues attack the bacteria, and within the first hour, neutrophils now invade the infected area. Okay, now invade the infected area. Okay, thus the macrophages are the first line of defense against bacterial infection, and while the neutrophils are the second line of defense. So the first line of defense against bacterial infection is what? Macrophages. Why the second line is what? Um, why the second line is what? Neutrophils. Okay, neutrophil. So an infection is associated with what? With a marked increase, a marked increase in the number of neutrophils. And this is what we call neutrophilia. Okay, that is an increase in the number of neutrophils in blood. We call it what? Neutrophilia. So if you did a blood count and you saw that the number or the amount of neutrophils in blood is higher than usual, you may begin to suspect what? Bacterial infection. Okay, you begin to suspect what? Bacterial infection. Okay, so this neutrophilia is as a result of what? Stimulation of bone marrow to release large numbers of neutrophils. Okay, and what stimulates this is by certain products of inflammation enter the bloodstream and cause this and stimulate the bone marrow to release more neutrophils. Okay, so now the neutrophils are in blood. How do they reach the site of infection? That's very important. Please, you need to know this. How do the neutrophils reach the site of infection? How do the neutrophils reach the site of infection? So the first thing that the neutrophils does is what they are first attracted to the capillary wall. Okay, they are first attracted to the capillary endothelial surface by proteins called selectins. Selectins. So the neutrophils are first attracted to the capillary endothelial surface by proteins called what? Selectins. And then they roll along it. Okay, this rolling of neutrophils on the capillary endothelial surface is what we call margination. It's called what? Margination. Okay, margination. Okay, so the first thing is what? The attraction of the neutrophils to the surface of the capillary endothelium by proteins called selectins. Okay, now when they are attracted, they now bind. These neutrophils now bind firmly to a protein, to what we call adhesion molecules, and the protein to which they are bound to is called integrin. Integrin. Okay, so they bind firmly to a protein. Okay, to a protein adhesion molecule. Then when they bind to it now, they now insinuate themselves through the capillary walls. Okay, you know, the capillary walls have the capillary have walls. Okay, so they insinuate themselves in between the capillary walls. Okay, between the endothelial cells by a process called diapedesis. 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 Okay, so the thing that happens that they, okay, well, after the diapedesis, they now actively move toward the site of infection by an amoeboid motion. Okay, so we talked about margination, then we have talked about what? Diapedesis, and then they now move to the site of infection by what? An amoeboid motion. Of course, we remember amoeba, right? Okay. But then they are attracted to this site by chemicals. Okay, they are attracted to this site, to this infected area, by many chemical substances, and this process of attraction, chemical attraction, is called chemotaxis, right? Chemotaxis, chemotaxis. Okay, and these chemicals are called chemotactic agents or chemoattractants. Okay, chemotactic agents. Okay, or chemoattractants. Chemoattractants. Okay, and chemotactic agents, chemoattractants, chemokines. Okay, and the most important are which are the most important of these chemoattractants or chemokines are one. We'll look at that complement system, component of the complement system. Then the second is polypeptides from lymphocytes, mast cells, and basophils. Okay, polypeptides from lymphocytes, mast cells, and basophils. Okay, proteins. These serve as chemoattractants. Then some bacteria or viral toxins, okay, and degenerative products of inflamed tissues. These are chemicals that will attract neutrophils. Okay, we say the first one are components of the complement system. The second is polypeptides from lymphocytes, mast cells, and basophils. The third is what? Some bacteria or viral toxins and the degenerative products of inflamed tissue. And then also have leukotrienes. Okay, leukotrienes. Okay, these are chemicals that will cause attraction. Okay, so we've looked at how the chemicals, this neutrophil, the process by which a neutrophil reaches the site of infection. But when it reaches it, what is the mechanism of action of neutrophils? Okay, how do the neutrophils act? Okay, first there is the process of called opsonization. Opsonization. Okay, there are certain plasma factors that coat the bacteria, making it tasty for the neutrophil to eat. Remember, we said it phagocytoses a microorganism and then destroys. So for it to phagocytose, the microorganism should be appealing to the neutrophil, so to speak. So there is first what we call opsonization, coating of the bacteria with plasma factors that make it tasty, that makes it a little bit appetizing for the neutrophil. Okay, so we have certain plasma factors coat the bacteria, making them tasty to the phagocytes. Okay, so this process is called what? Opsonization. And the main opsonins, and so these chemicals that coat the bacteria, we call them what? Opsonins. Opsonins. Okay, and the main opsonins are what? Complements of the components of the complement system and immunoglobulin G. Okay, immunoglobulin G. Okay. Lucas, if you don't mind, probably maybe can somebody help Lucas with that so that the class is limited, or you may want to play the tape again, but so that you catch up with us anyway. So we talked about, you said you ended with diabetes. So the first part we were talking of was how the neutrophils reach the site of infection. We talked, the first one was the margination, where they are attracted to the capillary endothelial surface by selectins. Then they now bind to the protein integrin, and then they now insinuate themselves through the capillary walls between the endothelial cells by the process called diapedesis. Then after they have gone from the capillaries into the tissues, they move towards the infection area by amoeboid motion. And then, but what is attracting them towards that site is what we call, what is attracting them are chemicals, okay? And this process is called chemotaxis. And these chemicals are chemokines, chemoattractants, chemotactic agents. And we listed the various chemotactic agents, components of the complement system, polypeptides from lymphocytes, mast cells, and basophils. And then we also have some bacterial or viral toxins, and degenerative proteins of inflamed tissues. Then we talked about leukotrienes, okay? So now we are looking at the mechanism of neutrophils, and we said the first thing, that's beautiful, Naomi and Inonge, so that's okay. So the first and as well as immunoglobulin G. Okay, let me make a correction. Naomi, opsonization now is not one of the process where the neutrophils reach the infected area, but when they reach the infected area, how do they destroy? So the first process is opsonization. So opsonization is the first mechanism for action of neutrophils, okay, not a process by which they reach there, okay? So make that correction, Naomi. So then the second process, after they have coated the bacterium or the bacteria, the coated bacteria now binds to receptors on the neutrophil cell membranes. So the coated bacteria now binds to receptor, on the neutrophil cell membrane. Okay. Now the coated bacteria is now ingested inside these cells, and by a process of endocytosis. So there is ingestion of this coated bacteria. Remember, it is first bound to receptors, and then it is taken in, okay? It is phagocytosis. It is by the process of endocytosis, okay? And then it forms, when it is endocytosis, it forms phagocytic vacuoles, okay? What we call phagocytic vacuoles, okay? Phagocytic vacuoles, okay? And then these vacuoles that contain the phagocytosed bacterium are called what? Phagosomes, okay? Phagosomes. Okay, so the first thing is what? Opsonization. The second one is what? Phagocytosis. Now, and this bacterium is phagocytosed and then that's endocytosed, and the phagocytic vacuoles are formed, which are called what? Phagosomes. Okay, now certain cytoplasmic granules now come in contact with these phagosomes. First, the bacterium is opsonized. Secondly, it binds receptors on the neutrophil, and then it is being endocytosed and forming phagosomes. Then inside the neutrophil, some cytoplasmic granules now come in contact with the phagosomes and discharge their contents into the phagosomes. Okay, so the first thing is what? Opsonization. Second one, phagocytosis. Phagocytosis, forming what? Phagosomes. And then the next thing is that what? They have what? Cytoplasmic granules. Cytoplasmic granules come in contact with the phagosomes and release what? And release their content and discharge their content. Okay, so this process is called degranulation. Degranulation. Okay, degranulation. So the cytoplasmic granules empty their content into the phagosomes. Are you following Kinley? It's quite important. Okay, now what are the issues of this? These granules contain what? What is the form? What are the content of these granules? Okay, they contain proteases. They contain proteases. The content of the granules, one, proteases. Okay, these are, when we say proteases, those are proteolytic enzymes. Okay, these are proteolytic enzymes. Proteases are what? Proteolytic enzymes that digest what? The bacteria. Okay, then the second content of the granules on the what microbial proteins called defensins. Okay, so we talked about opsonization, talked about phagocytosis, formation of phagosomes. Then the cytoplasmic granules come in contact with these phagosomes and discharge and degranulate their content. So we are listing the contents. We have proteolytic enzymes that digest the bacterium. We have defensins. Okay, then we have myeloperoxidase. Myeloperoxidase enzyme. Okay, myeloperoxidase enzyme. Okay, these enzymes catalyze the reaction between hydrogen peroxide and chloride. Okay, and chloride. Okay, so this myeloperoxidase enzyme catalyze the reaction between hydrogen peroxide and chloride to form what? Hypochlorite. Okay, and this hypochlorite is a powerful and bactericidal substance, so it kills bacteria. So this hypochlorite is what? It's bactericidal. We call it bactericidal. Okay, then it is called suicide of the bacteria, right? It's suicidal to the bacteria, so it's called bactericidal. Okay, so so the first opsonization phagos, degranulation, and then we've talked about. Then the next is what? Then we talk about they have the next one is NADPH oxidase, NADPH oxidase. Which is formed by the organelles called peroxisomes. We saw that last peroxisomes. Okay. The NADPH oxidase is formed by peroxisomes, and then they are bound they are bound to the phagosomal membranes. Okay. So these NADPH oxidase are formed by peroxisomes and are found on the phagosomal membrane, and during this process they are activated. Okay, so Mukuka determine declarations when cytoplasmic granules come in contact with phagosomes and release. Yes, that's right. Okay, so this enzyme, this NADPH oxidase, catalyzes the formation of superoxide. Okay, superoxide. Okay, so it catalyzes the formation of superoxide from free radicals. Okay, this NADPH, catalyzes the formation of superoxide. Okay, which is a free radical. Okay, which is a free radical, and so the superoxide reacts with hydrogen ion to form what? Hydrogen peroxide. So the superoxide reacts with what? With hydrogen ion to form what? To form hydrogen peroxide by the activity of the enzyme superoxide dismutase. So we said the NADPH oxidase, what does it do? It catalyzes the formation of superoxide, and this superoxide combines with hydrogen ion to form what? Hydrogen peroxide by the action of the enzyme superoxide dismutase. Okay. And please, I've still said it again. Please learn to ask questions. What is the function of defensins container? No. Ask your question in a polite and honorable manner. Okay. Please, what is the function of neutrophils? If you're directing it to me, you direct it to me. If you're directing it to the class, someone will help you answer. But don't be disrespectful and arrogant and verbose in a manner that is incoherent with ethics of communication. Okay, ask your question politely. What is, okay? So the superoxide and the hydrogen peroxide that is formed are oxidants and are effectively what bactericidal agents. Okay, there are also what there are bactericidal agents. So we need to understand that. Okay, so they help in killing the bacteria. So let's look at eosinophils. Okay, eosinophils, just like neutrophils, have a short life in circulation. We mentioned about 4 to 6 hours, and are attracted to the endothelial cells by selectins, and they bind to integrins, and then they enter the tissues by diapedesis. Okay, and they are actually abundant in the gastric, in the GIT mucosa. Okay, gastrointestinal tract mucosa. Okay, they are abundant in the GIT mucosa. Okay, where they defend against parasites. Now, I told you when you hear of neutrophils, what comes to your mind? Bacterial infection. When you hear of eosinophils, the first thing comes to your mind is what? Parasitic infestation, okay, and allergic diseases. That should be in your mind, okay. Somebody say I'm too fast. Which one? I think I'm taking it over and over and too slow. Okay, so eosinophils, they follow the same. While I rushed with eosinophils because they follow the same process like neutrophils. Okay, by attaching to endothelial cells by selectin, bind to integrin, and then enter the tissues by diapedesis. It's the same process. Okay, and I'm now saying that they are abundant in the GI mucosa, where they defend against parasites. Okay, as they are also present as well in the respiratory and urinary tracts in abundance. Okay, they are present in the abundant in the what, in the urinary tract, in the respiratory tract, okay, and the GIT mucosa. Okay, so they are also phagocytic and they exhibit what, chemotaxis, but they are less motile and weaker than neutrophils. So they follow the same process. Okay, so they follow the same process. Okay, they follow the same process. Okay, so that's quite important. So they follow the same process. So, but then is that the difference is that they are less motile than neutrophils, and then they are weaker. Okay, but then I said what, they are abundant in what, in the GIT mucosa, in the respiratory tract, and the urinary tract. Okay, and what use infections. Okay. Okay, so they are produced in large numbers in conditions of what, parasitic infestations. Okay, in which the eosinophils now attack the parasites and kill them by hydrolytic enzymes. Okay, the process is almost this is almost similar. Okay, and another thing is that they are produced in large quantities in allergic diseases like what, bronchial asthma. Okay, bronchial asthma, they are produced in what, in large quantities. Okay, in large numbers. Okay? So when, if you have eosinophilia, you start suspecting either parasitic infestations or the person has what, allergic diseases. Okay, so in this allergic disease, the eosinophils are attracted to the sites of allergic reactions by chemotactic factors that are released from basophils and mast cells. Remember, I mentioned to you that they are abundant where, in the GIT mucosa, in the respiratory tract. And if you look at in these places, we have what we call enterochromaffin. For example, in the GIT, we have enterochromaffin-like cells that resemble mast cells and basophils. And then in the respiratory tract, we also have mast cells and basophils, so to speak. Okay, and these substances, these cells, okay, and then we also have eosinophils. Okay, we also have eosinophils. So these eosinophils react in issues of what, allergic reactions. Okay, and so, and they are attracted to the site of disturbance or infection by chemotactic factors that are released by basophils and what, mast cells. Okay, and we'll look at that briefly later on. Okay, so here they attract to the site of infection where they decrease and prevent what, the spread of the allergic inflammatory process. So how do eosinophils prevent the spread of allergic inflammatory responses or processes? One, by detoxification of some inflammatory inducing substances. Okay, so they detoxify some of the inflammatory or inflammation inducing substances released by basophils and mast cells. Okay, so I remember I mentioned to you that we have basophils and mast cells that are abundant in the GIT and in the respiratory tract, and they release pro-inflammatory chemokines. Okay, that means they release chemicals that promote inflammation. So the eosinophils would also release substances that detoxify, or they try to detoxify some of these inflammatory chemicals that are released by basophils and mast cells. They also phagocytose and destroy allergen antibody complexes, because the allergen can they can be bound by an antibody, and then what, this complex is called the allergen antibody complex. Okay, and the neutrophils phagocytose this complex and destroy them. Okay, so let's look at basophils. Basophils are non-phagocytic cells. They are non-phagocytic cells that resemble mast cells. Okay, basophils are non-phagocytic cells that resemble what? Mast cells. Okay, basophils are what? Non-phagocytic cells that resemble what? Mast cells. Okay, and both mast cells and basophils release heparin and histamine and other inflammatory mediators, what you can call what? Leukotrienes. Okay, so we have basophils and mast cells release what? Histamine, heparin, and other inflammatory mediators like what? Leukotrienes. Okay, leukotrienes. Okay, leukotrienes. The other day, we were discussing about formation of prostaglandins. Okay, and we talked about being formed from phospholipids to arachidonic acid, and from arachidonic acid we can form prostaglandin E2, prostacyclin, thromboxane A2, or we can even form prostaglandin prostaglandin F2 alpha, and we can also form what? Leukotrienes. Okay, leukotrienes. So these leukotrienes are inflammatory mediators. Okay, and they own both bradykinin and serotonin. So what are the chemicals? The first thing, the first assignment I want to give the morphological and functional similarities or differences between mast cells and basophils. Okay, the functional, the structural similarities and differences between mast cells and basophils. Okay, so that is the first assignment. Okay, the structural and functional differences between mast cells and basophils. Okay, so that's important. Okay, so these substances that are released produce what we call they produce the local vascular and tissue reactions that occur in allergic conditions. Okay, so they produce local vascular and tissue reactions that occur in allergic conditions, okay, and are responsible for what we call the immediate type hypersensitivity reactions. Immediate type hypersensitivity reactions. Okay, so that they're involved in that. So okay, these substances are involved in they produce a local vascular and tissue reactions, okay, that occur in allergic conditions. Okay, and are responsible for the immediate type hypersensitivity reactions. Okay, and which may be as mild as urticaria and rhinitis, or as severe as what? As anaphylactic shock. Okay, Makowani. Okay, I said the structural and functional differences okay, and similarities between mast cells and basophils. Okay, between mast cells and what? Basophils. Okay. Okay. So we've looked at that. So let's look at monocytes. Monocytes, we said monocytes, they have a lifespan of close to, they're about 70 hours or so. And we said they enter the blood tissues and become tissue macrophages, okay? They enter the blood and become tissue macrophages, okay? So they make the monocytes enter the tissues and become what? They leave the blood and enter the tissues and become what? Tissue macrophages, okay? And they are the main constituents of the reticuloendothelial system, okay? So monocytes are the main constituents of the reticuloendothelial system, and they offer the first line of defense against bacterial infection. Okay, because when they enter the tissues, they become what? Tissue macrophages, and they offer the what? The first line of defense against bacterial infection, while neutrophils offer what? The second line. Okay, so in cases of severe inflammation, they are released into the bloodstream from the bone marrow in large numbers. Okay, then they move to the site of infection by diapedesis, amoeboid motion, and chemotaxis. The same process like the neutrophils. Okay, so in severe infection or in severe inflammation, they are released from the bone marrow into the bloodstream, and then they move to the site of infection just in the same manner in which neutrophils move. Okay, yeah. So such process will require a few days. Okay, and it's quite slower than the movement of neutrophils. Okay, and so in addition, monocytes engulf what? Necrotic tissue and play an important role in immunity. We'll look at that. So they engulf necrotic tissues that have experienced necrosis, cell death. Okay, so where they are necrosed. Okay, so we have lymphocytes, and then lymphocytes are the major players of the immune system. So we'll look at that. Uh, So the second assignment which you have to do is what are the variations of the leukocyte count? Variations, that's the physiological and the pathological variations, okay? The physiological and the pathological variations of leukocyte count. So let's quickly look at the endothelial system. What is the reticuloendothelial system? Okay, we also call it the tissue macrophage system. Tissue macrophage system. Okay, or what? The reticuloendothelial system. Somebody's blocking this thing. Check if you're getting me, please. I've sent comments and unable to connect to chat. Please try again later. Please, if you can get me, let me know. So, but then comments are not coming up. Sir, can you send a comment somewhere? Let me better see. Send a comment. In any case, I'm talking about the reticuloendothelial system. Okay. Okay, this system consists mainly of the tissue macrophages, which are originally monocytes that have entered from the blood. Okay, and we have a few specialized endothelial cells too in the bone marrow, the spleen, and lymph nodes. Okay. Have you seen a comment somewhere? It's not showing.
They're loading.
Huh?
But they're loading here before.
You are seeing comment?
Mm-hmm.
I'm not seeing anyone here. Just a moment, please. Comments are not coming on. was here. Thank you. Somebody has blocked their computer comments. So we have this system mainly involves the system consists of the tissue macrophages, which were originally what the monocytes that have left the blood and have entered into the tissues, and we have few specialized endothelial cells in the bone marrow, the spleen, and the lymph nodes that also are part of the reticuloendothelial system. Okay, and what is the function? What are the functions of the reticuloendothelial system? The first one is what? Defense. Defense. Okay, the first one is what? Defense. Okay, but can you see my comment there? So the first one is defense. Okay, so the tissue macrophages attack and phagocytize or phagocytose the invading organisms in the following sites. Okay, in the skin, these macrophages are called histocytes. Okay, in the skin they are called what? Histocytes. Is who has done this? Unable to connect to chat. Please try again later. I can't be able to spell out some things I needed to. Hey, please, if you follow me and you've manipulated something, please make sure you try to undo it. So in the skin, they are called what? Histiocytes. Okay, in the skin and the subcutaneous tissue, they are called what? Histiocytes. So they help in mopping up any microorganisms of the one that enter through the skin. In the lungs, they are called pulmonary alveolar macrophages. Okay, in which bacteria and foreign particles like dust, carbon and silica are being what? Are being engulfed. So they are called what? Pulmonary alveolar macrophages. Okay. In the liver, they are called what? The Kupffer cells. Okay, the Kupffer cells. The Kupffer cells. And so they are able to attack bacteria that continuously pass from the GIT. Probably maybe you ate some stale food, what kind of food you've eaten, stale bread and things, and some microorganisms succeed in entering the blood system. First of all, passes through the liver, and then the Kupffer cells engulf this. So in the lymph nodes, they also attack bacteria transmitted from tissues. Then in the bone marrow and the spleen, they are able also to attack bacteria that enter from the bloodstream. So in the bone marrow, they are called what? Osteoclasts. Okay, they are called osteoclasts. I think sometimes ago I discussed this with your class, if I'm not mistaken. And so the second function is what? Removal of old blood cells. Remember when we're talking about degradation of red blood cells, erythrocytes? That when they are old, they are picked up by what? By the reticuloendothelial system. Okay, so they remove what? Old blood cells. Then the third function is of the reticuloendothelial system is that they break down hemoglobin and form biopigments as well as store iron. Okay, so they break down hemoglobin, they form biopigments, and they store iron. What are examples of? I can see your comments. So we have what? Bilirubin and biliverdin. Okay, bilirubin, biliverdin. Okay, so they also repair damaged tissues after inflammation. Okay, they repair damaged tissue after what? After inflammation. Okay, by releasing tissue growth factors. They also serve as extra medullary hemopoietic sites. Remember when we were talking about blood formation? We said they are formed from the embryonic stage, the extramedullary stage, and the medullary stage. Okay. Then the last function was erythropoietin. Some erythropoietin is formed from the Kupffer cells of the liver. Okay, from the Kupffer cells of the liver. Okay. And then so let's look at functions of spleen. Functions of spleen. Functions of the spleen. Okay. We said the spleen also forms part of the lymphoid organs. Okay. So the first function of the spleen is what? Defense. Okay. Okay. It produces large numbers of macrophages in the spleen. So yeah, the spleen produces large numbers of macrophages. It helps in the formation of lymphocytes and plasma cells, which play a role in immunity. The spleen has been removed. Okay, if the spleen has been removed, you can imagine, right? The person is prone to bacterial infections. So the second function of the spleen is blood cell formation. Okay, red cells in the fetus and lymphocytes in the adults. Red cells in the fetus and lymphocytes in what? In the adults. Okay. Then the third is what? Blood cell removal. Old and abnormal cells are removed. Okay. What are the kind of abnormal cells we are talking of here? Spherocytes, okay, elliptocytes, okay, sickle cells, okay, they are removed by macrophages in the spleen, and then the hemoglobin is released and undergoes the same process we discussed before. Okay. And then we also we could also have instances where you have excessive platelet breakdown in the spleen, okay, which could cause thrombocytopenic purpura. I think I gave you the notes, right? Yeah. So also the spleen also helps in blood storage, okay. But then the human spleen can accumulate just about 50 mL of concentrated blood in its sinuses, okay. And so this blood can be added into the general circulation, by contraction of the smooth muscles in the splenic capsule, okay, during excitation of the sympathetic nerve supply. Okay, so when we have issues, for example, during hemorrhage, okay, after hemorrhage, the sympathetic nervous system is activated, and they can stimulate the smooth muscles that are present in the splenic capsule, and this will lead to contraction of the spleen to release this 50 ml. But this is sometimes it's not important because the quantity is small, and the contraction of the splenic capsule sometimes is relatively weak. So imagine releasing 50 ml into the circulation when somebody has lost like a liter or more. But in as much as this still counts, okay. So let's quickly look at immunity, okay. But before we look at immunity, please permit me to rectify the common things, because it will not be teaching if I'm just talking and going. Okay, so I will disconnect and connect again. Okay, thank you, please. So stay put. Someone, can you check this?